Welcome, Dr. Stalin. You have 20 minutes, please.
Well, good morning, and I'm going to be talking about solvatochromic dyes. I'm going to race through the first half of the presentation, and it's going to be like a recap of presentations I've done before, so we can then focus more on recent results. The dyes are producing a wealth of information, and there are more questions than answers. I mean, every experiment produces an answer, but 2 or 3 questions, which I suppose is how research is supposed to go. If there are any chemists in the room who'd like to come up to me afterwards and provide any input on what will be, I suppose, basically a chemical presentation, that would be very welcome. So let's just start and go through these. So why use solvatochromic dyes? Basically because they're very sensitive to all kinds of things. And so if there are any changes in solution induced by potencies, they're likely to pick those changes up. It also turns out that there are a whole range of solvatochromic dyes that can be used under different conditions. So they give slightly different information. They all share a common structure, which is electron donor, electron bridge, and electron acceptor. And for those of you who like chemical structures, the top 4 are negatively solvatochromic dyes, which means their spectrum blue shifts as the solvent becomes more polar, and the bottom 4 are positively solvatochromic dyes, which means their spectra redshift as the solvent becomes more polar. That's just a diagram showing basically what happens. The bathochromic is red and hypsochromic is blue. So the first paper was really just showing that these dyes do detect potencies. And the only figure I'll lift out of that paper is this one. And really I've done it to show that the potency effect rapidly reaches a maximum and then slowly declines. And we'll see this again later in other studies. And the kinetics, basically kinetics is a process over time, is a really big key as to what's going on with potencies. So the second paper was using a water-soluble dye, BDF. And here, the only slide I'll show you is that at high pH, there's a mechanism whereby the dye decomposes, and potencies stop that or inhibit that decomposition. And it's actually to the extent that it's visible to the naked eye. So after a few days, the control solution is almost colorless, whereas the potency solution is still quite pink. And this was the first time that it became clear that something strange was going on, and that potencies seemed to be inducing some kind of electron density shift, which then inhibited this decomposition. The next paper then was looking at a whole lot of different solvatochromic dyes, and just to show you that there's some sort of correlation between the degree of effect of a potency and the dipole moment at the ground state of the molecule. I think it also applies to the excited state, for those of you who are familiar with the way electrons move, but it doesn't correlate with the transition dipole moment. Why, I don't know, but it's an observation that needs to be clarified at some point. Just 2 of the compounds that were looked at, Brucher's myricyanin and diaminonaphthalene disulfonic acid, produce some interesting results. I can't go into detail because of the time constraints, but basically, in the presence of potency, as pH goes up, the potency further encourages the loss of a proton from that form of the dye, but at low pHs, it encourages the protonation of the dye. And furthermore, it seems to encourage the transformation of the trans form to the cis form. There's no common mechanism for all of that. It's a real conundrum, but one possibility is that if a system's moving in one direction, potencies move it further in that direction. If it's moving in the other direction, the potencies move it further in that direction. And that's sort of reminiscent of clinical practice, that on giving a good remedy, that you get exacerbation of symptoms. So maybe this is mirroring that in a chemical system. That's if you encapsulate with a beta-cyclodextrin, then you don't get those effects. And what you see is the effect of how the encapsulator is interacting with the dye. And so you don't see the trans-to-cis isomerization, and you only see protonation, which actually isn't. It's the cyclodextrin binding to the dye. And then with this other compound, again, you see a very big difference between controls and potency solutions. So there's some sort of what we think is an intermolecular reaction, which is inhibited by the potency. And I think it's because what actually happens is that potency promotes protonation, and then that stops that intermolecular reaction going on. So there's some big differences which you can exploit. And the next paper is looking at how, when you do encapsulate these dyes, what happens? Because when you encapsulate, that's just to remind you of the difference between positively and negatively solvatochromic dyes. So with positively solvatochromic dyes, you get a redshift in the wavelength if the solvent becomes more polar or there's an applied electric field. The opposite is true with negatively solvatochromic dyes. The thing about encapsulating is that it stops the dyes aggregating, it stops them protonating, and it stops the solvent getting to them. So if you see any changes, it has to be due to an electric field. And with 4 different positively solvatochromic dyes, you see in every case a red shift in the spectrum. All of these show a red shift. And conversely, with negatively solvatochromic dyes, 4 of them, they all show a blue shift in their spectrum. So they're all responding to some kind of electric field. Now, for Brucher's mericinin, there's both solvatochromic and electrochromic data available, which means that you can take the shift in the spectrum seen with a potency and then correlate that with what sort of electric field there might be associated. And in this case, it's arsenicum 10M. And it turns out that the figure that drops out is about 10 to the 7 volts per meter. And that is comparable with the potential difference across cell membranes. So that's probably enough for potencies to have an effect electrically on systems. And it's a nice experiment that it shows that the potencies do have an electric field. So the next paper was a beginning of a collaboration with Professor Bonomin at the University of Sao Paulo. And I just want to show this slide, which shows that with the dye ET33, as the potency level goes up in antimonium crudum, you get progressively increasing effect. If the potencies are subjected to a weak electric current, it gets knocked out completely. So this is paradoxical, that the potencies seem to be producing an electric field of their own, but if you pass an applied electric current through them, it knocks them out. The next slide, again, the same paper, antimonium crudum. This is the effect of the potencies on methylene violet. If you look at, if macrophages have those potencies sort of pass through them, and what happens is the macrophages amplify the potency effect. The big question is this a universal feature of living systems, that they can amplify potencies? And it's something that we don't know yet, but it's a very interesting result if it's correct. And then the next paper, again with Professor Bonomin, is looking at how solvatochromic dyes pass through large bodies of water. And in this case, it's a lake system of 55 million litres, which was seeded at the source with 1 litre of phosphorus 30. And within minutes, the whole lake system turns into phosphorus 30. Now, if you, I mean, these are the, this is the data. So this is the beginning of the lake system, and you can see how it builds up to a maximum and then declines. Again, it builds up to a maximum and declines. And this is the last lake in the system. So it's gone from the beginning to the end in a matter of minutes. And if you just plot out the data, and there aren't many points because the experiments weren't designed to be plotted out in this way, but you can see that basically the entrance to the lake system and the last lake in the system become phosphorus 30 almost at the same time. So there's a very rapid increase in potency action, a plateau, and then a slow decline. And what might be the mechanism of the propagation of phosphorus 30 through this lake system? And the propagation is faster than the flow of water. So it's an interesting question, which I'm not going to answer just yet, because I'm going to divert slightly to one of the latest projects is to immobilize dyes on transparent cellulose films, which makes it much easier to study the action of potencies on the dyes. The main reason for that is that the dyes then can't aggregate, which spectroscopically complicates things when they do. And there's just an example of some of the dyes that have been immobilized, so there are a nice range of colors. And I'm going to focus on Brucher's merocyanin, which is this one here. And so the Brucher's merocyanin has been coupled to epoxy-activated cellulose. You don't need to know all of that, but it's a recognized method of doing things. And this is the change in absorbance over time for immobilized dye with arsenicum 10M. The controls, nothing happens for a while, and then that also starts to much more slowly decline. And if you plot out the difference between the arsenicum and the control, you'll see a familiar pattern. That's a very rapid increase in potency activity, followed by a plateau that lasts several days, and then a very slow decline. And we've seen that before. Oh, no, I won't do that. So that slow decline looks familiar if you're a chemist. It looks like a first-order rate process. And in fact, if you plot out the natural log of the difference in absorbance between immobilized arsenicum, Brucher's merocyanin, and the control, you get a straight line. Now, the other very common first-order decay in nature is radioactive decay, and I'm not suggesting that there's a comparison between radioactive decay and what's happening here, but first-order rate constants are quite common in nature. Quite a lot of things decay according to first-order kinetics, and what it means is that arsenicum is a discrete coherent agent. It's not some sort of diffuse cloud in the sky. It's actually something that's coherent, and once it's interacted with the target system, it starts to fall apart. So just to remind you, we've seen this pattern before, a very rapid increase in activity, a plateau and a slow decline. With the lake system, it was the same, a very rapid increase in activity, a plateau of over several hours, and then a slow decline. The timescales are faster in the lake system than they are in immobilized dye. I don't know why that is. In the paper, I offered an explanation, but to be honest, I don't know. It's one of the many questions that we have. And just to go back to one of the first slides, you see the same thing. So a rapid increase and a slow decline. So three totally different systems produce roughly the same result. That is, the potencies are increasing in activity, staying relatively stable, and then they decline after they've interacted. which sort of makes sense on a clinical level, that when a potency is given to a patient, they don't go on reacting forever. I mean, there's a period of reaction and then a decline in that interaction. So that, again, sort of makes sense. So what do I think is going on? I've talked about the decline phase. The plateau phase is a sort of interaction phase where the potency is slowly being exhausted. But what about that rapid build-up phase? Well, I can't give you the evidence, but I think that potencies are subject to fractal growth, that in solution they spread by fractal growth. And fractals are everywhere in nature. I mean, there are so many different examples. I mean, all of these are all examples of fractal growth. This is a chemical system of copper crystals, how they grow, and we saw that yesterday in Stefan's talk. So why not potencies? So what evidence is there? So the types of responses to dyes suggest that initial conditions are important for potency growth in solution. And that's very characteristic of fractals. The rate of spread of phosphorus-30 in a lake system is faster than the flow of water, so the mechanism of spread must therefore be by rapid growth and self-replication, independent of water flow. I mean, a simple thing is that potencies are inexhaustible and infinitely divisible. In other words, they grow to fill the solution in which they're put, and any portion is the same as a whole. So you take a portion and add water, and it grows to fill the volume again. You take a portion, it grows to fill the volume again. And every portion is the same. So again, that's very characteristic of fractals. And the last sort of piece of evidence, circumstantial admittedly, is that standard deviations for potencies tend to be larger than for controls, indicating an inherent variability in potency strength. And that's something I think we've probably all ought to be aware of, that potencies aren't static, and they do vary in strength over time. If we had more time, I'd give you some examples of why I think that is, but let's move on. So the next question is, what role is water playing in the potency, propagation, and action? I mean, is water intimately involved in the propagation, or is water solely an inert carrier? So the first step in answering these questions is to find out if pure water can be potentized. If it doesn't potentize, it's possible or likely that water is simply an inert carrier. So comparing controls, water 10M and arsenicum 10M, using the Pinker's potentizer, Tony Pincus from Ainsworth Pharmacy has developed the Pinker's potentizer, and using the solvatochromic dye phenol blue. So this is the potentizer. Samples, cells A and B, are made from 304 stainless steel, which is highly resistant to ultra-pure water. There's a consistent downward succussion force of 3G. Cells are baked at 200 degrees for 1 hour between succussion runs. There's a mu metal barrier between cells, and it's essential that water 10M and arsenicum 10M are kept separate at all times. So there we are. There's water, there's succussed water, and there's arsenicum 10M. That's using cell A. Switch round, and you get the same result, which suggests that water itself can't be potentized. It's only what is put into the water that becomes potentized through dilution and succussion. So water appears to be an inert carrier. So in conclusion, what can we say about homeopathic potencies from the use of these dyes? Potencies act to polarize these dyes. They act in a general manner to accentuate equilibrial displacements, and we've seen that with several of the dyes. Arsenicum 10M at least, and probably all potencies, possess an electric field, but a weak electric field applied to the potency neutralizes it. Macrophages and other living systems amplify the activity of potencies. Phosphorus-30 spreads through a lake system faster than the flow of water, indicating fractal growth. Other kinetic evidence supports that. Arsenicum 10M loses its activity according to first-order kinetics, indicating potency of the discrete coherent agents, and water itself can't be potentized. It appears to be an inert carrier. So there's a lot we can say about potencies, but we still can't say what they are. But we're getting there. it's a slow process. That's science, really. Thank you for all the people that have supported this research over the years. It wouldn't have happened without their help. So thank you.